This talk is not about all the stuff we did this summer. We did have a good summer. My summer got really interesting in August. I went on a cruise with my dad and his girlfriend and my wife and 20-something, 40-something other people overall from my dad's retirement center. We went up to Alaska and saw Glacier Bay and good weather on Glacier Bay. We also stopped at right across from Glacier Bay. There's a Rocky Icy Point, which is a tourist stop just for cruise ships. And they swear, they're looking at the geology, that that's a Silurian tempestite deposit along the beach there, or turbidite deposit. But I think it's all Silurian stromatolites myself. But they wouldn't let me bring any back on the boat. So I didn't do much fossil hunting then. Turned out my dad had a bad valve in his heart. He couldn't walk very far on the ship. So we came back, and in the 5 days between when we got back from the cruise and when I went up to Canada, we had him in the doctors. He got diagnosed as having a bad heart valve. They did a type of surgery on him when I was up in Alaska, up in Vancouver Island. They went in through the femoral artery and they replaced his valve, closed heart surgery. And he is doing marvelous. He went home the next day. He's doing great. Doesn't have any shortness of breath, no loss of energy. He's still not exercising like he should, but that's okay. We can all live with that. He's 97 and a half. If he doesn't want to have to exercise, I can't make him. And then we went up to Alaska, or to Vancouver Island, and went on several field trips. We had a lot of great presentations by the folks up there. A lot of paleontology is available just within British Columbia itself, let alone other places. There's some interesting stories that are developing. We think that, what the insular terrain, which is where Vancouver Island and some of the associated stuff on the coast originally was deposited and formed as rocks off of Baja California and migrated north and got spread along the coast, and all sorts of interesting stories. That's not what I'm going to talk about tonight. So I'm going to talk about bones and fossil bones. This whole thing has risen. Go ahead and go to the next slide. Yeah, has risen. Yeah, okay. Out of a question that one of my visitors at OMSI asked me I was talking to him about Bernie's bones, Bernie the thalattosaur. They asked me a simple question: why are the bones black? Now, the glib answer is, well, they get stained by minerals in the rock. But I don't think that's the right answer. So I am starting an investigation to determine why these bones are black. Actually, in thin section, if you look at them in thin section, which you can't see from down there, but I can see just fine, they're brown. But still, why are they not bone colored? Well, so I started looking at organics in bones, and also basically what is the fundamental structures of bones. Bones themselves are derived from early strengthening structures found in Cambrian and maybe even Precambrian organisms. They are found in things like Pachia Crasianus, which is from the Burgess shale, and other things like that, which are basically structures made by the protein collagen, or collagen, however you want to pronounce it. Collagen is a rather strong protein arrangement. You makes nice spirals, and the spirals link together, and the linked spirals link together, and it makes up spiral and rod-shaped structures in your body, and it's found in bone and in cartilage and in many other parts of your body structure. And the difference between cartilage and bone is just that you have collagen there, and in bone, there is also co-deposited within the collagen, there are mineral aggregates of calcium phosphate or calcium hydroxyapatite. Calcium phosphate with a few OH groups stuck into it. So bones are basically strengthened cartilage, which is why it makes sense that animals that have cartilage for their bony structure, such as, skates and sharks and things like that, it's the same structure. It's just one has minerals in it and the other one doesn't. Bones change during their growth and with time. Of course, bones have to grow and add more as organisms grow. Juvenile bone looks different. It's laid down more quickly. It has a more disorganized structure than mature bone, which is very interesting for telling when we talk about Bernie. They have, as they grow, if the animal goes through periods of less food, they can develop lines of arrested growth, which are generally correlated to annual growth periods. But as you get to be an adult, your bone gets reworked all the time. It's continuously being destroyed and reworked, rebuilt along this mechanical stress line. So the bone structure, the cells can sense mechanical stress, and they build the mineral structures based on mechanical stress the bone is experiencing. So as you get older, or as you go through time, what you're doing with your body changes, and the bones get rebuilt. That in old bones, mature bones don't have lines of arrested growth, for example. So we don't know how old a T-Rex was when it died, but we do know young T-Rexes how fast they grow, as an example. Bone is really complex. It's very heterogeneous. It means it's got a lot of different structures in it, both on the macro scale, mesoscale, and micro scale, all different scales from the atomic level on up. Next, please. So, macro bone structure. If you're bored, I'm sorry. Macro bone structure is, what does a bone look like overall? You've got, this long bones are the ones that they typically show because, they're easy to understand, and we've all chewed up on ribs and things like that. So they have dense bone around the outside, generally around the outside of the bone as a whole, because that gives us the best mechanical strength for the least amount of material. That's just the way that physics or mechanics or mechanical engineering works, is that if you've got 100 grams of bone that you want to make, the most efficient way to make it is distribute it in a form of a hollow cylinder, and then that gives you your most bang for your buck, the most strength for the mass. So we have dense bone around the outer side. That leaves the inside free to be doing lots of different things. Down on the ends, in a lot of places, we have kind of a framework that provides more strength for the ends of the bones and stuff. Little filaments of bone, what we call spongy bone. In the center not much goes on necessarily. So other kinds of cells can live inside the bone, particularly early animals developed bone cells or developed cells that will make white and red blood cells are made by cells that live inside the bone and called the marrow. They also use it for mineral storage and fat storage and things like that. Bones, as they grow, they don't grow on the ends where the cartilage of the joint, the sliding joint is. They instead grow back away from that point. Epiphyseal line, I think is what they call it. So it grows, the bone actually grows, and it's a really crazy process actually, back away from it. And then when the growth stops, those lines seal up, they bond together, and of course they get reworked, so there's no trace of them in an adult bone. That's how come when we're preparing bones, if they've got those little discs and things like on backbones or they've got these lines, you can tell it was not a mature individual. Growth plates, yeah, in vernacular English. Okay, there's holes where the blood vessels run in and out, of course. Okay, next. When we get down to the the middle structure, bones get really complicated. We have here, here's a cross section, and I'm sorry for you people online that it doesn't show up. But we have a cross section here of a long bone. You take a piece out of it, you look at it, you blow it up. It's got spongy bone on the inside, of course, because there's no need for bone structure in there. They have little tubes of mineral and collagen, thank you, complex that we call osteons. They have capillaries running in and out of them. In between, they have layers of basically a collagen mineral pudding that makes up layers, and the little dark spots that you see there, those are actually cells. There are cells that live within the bony structure, and they feed it and keep it alive, or at least and do the immunological stuff and keep things from eating it and stuff. Very, very complicated structure. Now, in immature bones, well, we'll get a picture of that, but this is drawn as if it is a fully mature bone and has been used for a long time and rebuilt. We also, in the spongy bone, they've got just kind of little tubes that branch and go between all the different parts. So in the mature ones, these osteons, these tubes, they have a laminated structure. It makes it stronger. In juvenile bone, they don't have that structure, and the bones are, although weaker, are also more flexible. So that's why young bones break but heal easier too. Okay, next. So here's a picture of a thin section of a bone. You got the central canal. That's the one that has the capillaries in it for feed and return. We've got the lamellar bone matrix. It's arranged in lines. We've got concentric circles, and then these are the osteocytes. That's the living cells that live within the otherwise solid bone structure, and they have little tiny connections, canali, that hook everything together. So these get actually fed their nutrients and their oxygen from bones here, or from the stuff here in the central canal. Next. Now, here's some pictures from Bernie's bones. This is, I got the slice here. Now, this is Bernie is the 230 million year old Carnian age Triassic thalattosaur that a number of us here in the group helped dig up in 2012. My daughter and I found it in 2011. From Central Oregon, it's the oldest bones found in Oregon. And these are some thin sections, and I am just amazed at how beautiful these bones are preserved. So this is taken in ordinary light in a microscope, and the brown stuff is all bone, and these little tiny dots in here, these are the osteocytes, the little channels and stuff. That's here. These are hollow spaces within the bone. This is the edge of the spongy bone to hard bone transition. Under this one, this is polarized light on the other side, and these are crystals. So I use polarized light because it highlights the crystals that are in the mineral crystals that are inside the bone. The bone itself does not allow or does not repolarize light very much, so it gets dark. And these repolarize the light. So these are all mineral crystals filling all these holes. Next. So here's kind of a repeat of the cycle, the size. We start out with the whole bone size. We look at the dense bone. We've got all these osteons in it. They've got the little canals in the middle that run the blood vessels up and down to keep everything alive. The laminar or lamella, which is basically sheet function. In there, if we look at it, it's collagen fiber. So this is all those twirled up pieces of atoms of organic collagen, which is a protein. They are bundled together. Each individual one has made up of little plates or sheets of collagen, and this is where the minerals are. So the minerals, the calcium phosphate, hydroxyapatite, they are inside here at the molecular level. And these little crystals that make up the thing, we'll explain in the next one, these are really small. There's like 100 atoms across. So we go from the full-size bone size down to atomic level. Everything is different as you go through bone. It's very complex. Bone itself is about 30 to 40% collagen by weight, about 50% by volume collagen. And the rest is, in living bone, of course, there's water in there, and there's also the minerals, inorganic, about a third inorganic. Okay, next slide. So here's a picture, some pictures of immature bones versus mature bones. These are animals that haven't, or that are not growing anymore, and their bones have been rebuilt by the destruction rebuilding process that go on in your bones. By the way, that's an old process because it goes all the way back as long as bones have been mineralized. So that type of rebuild process was probably going on back in the Cambrian or Precambrian as soon as animals started having collagen as their strengthening function. Probably predates the mineralization. But you see, so in mature bones, we have this nice laminar structure here. And in immature, they're kind of laid down quickly. They fill the thing in. As life goes on, they put more and more minerals in it, and those get worked and reworked according to the mechanical stresses present. So that's how come you can tell if a bone is from a immature individual or a mature individual. Oop, vice versa, yeah. Okay, next. I am not really sure. I think that's about 500 times. Pretty small stuff. So, what do we think? When I think of hearing that we have minerals in our bones, what comes to mind? What form is the mineral? So, what I thought bones were is like this sponge here, some sort of a crystalline structure uh, that the bones lay down and made the calcium and the phosphate in a nice crystalline structure. Turns out that's not the case. What bones look like is more like this pudding, where you have little bits of solid in a collagen mass. So you got a mass of the protein collagen. Think of cartilage with minerals in it. The minerals filling it in. That's more what bones look like at the atomic level. That's important when we talk about what happens when we fossilize bones. So next time you think of what your bone looks like, think of your grandmother's Jello pudding. Yeah, it's even got olives with pimentos. Yeah. Ooh, I can't. Okay. Next. Okay. What happens to bones after death? Well, the first thing is, of course, the living cells die off. They're no longer supported by the arteries and veins and things like that. Everything stops. Bacteria invade it, start eating it up, particularly in the center sections where there's the spongy bone, where there's lots of holes. They get in there, they start eating up all the living cells. That's the simplest thing. And that's what we find in stuff like Ice Age fossils. I think if we looked at these, we would find that same sort of thing here. Ice Age fossils, it's just the bone structure. And when you think of bone structure, it's actually collagen with minerals in it. It's the jello. And like the stuff we get out of the Yamhill River, those are bones where the living cells have died off, but nothing else has happened. The second thing is the open spaces in all the spongy bone gets filled in with minerals, and that by minerals physically washed into the openings of the bones. Bone breaks, the inside gets filled up with sand and dirt, and by minerals precipitating within the holes that are too small for grains to get into. But in any event, all the open parts get filled in with minerals. And last of all, the collagen starts getting broken down, and the part where the collagen was in between all the mineral grains gets replaced with some other kind of mineral. But as we have found out through my research, that often does not happen. Stage 3 is not as common as you would think. Most fossil bones don't get to stage 3. They stay at 2. So next one. Okay, so here's a picture of, again, one of these thin sections of Bernie's bones. That's this one right here. And it's a section of the backbone. And in the backbone, there's a great big hole where the spinal cord goes. So we have a big hole right here where was all filled in with mineral grains that washed into the holes in the big. We have here spots where so the first one is over here on this side. This is where the mineral grains washed in. You can see the size of them here. Over here, we have holes that are too small, and those minerals, they are minerals. You see they're colored. This is polarized light. Those minerals precipitated in place. And then lastly, we have the bone here. And that bone is basically still intact. So that means that half of this brown stuff you see here, half of this stuff is still the organic molecules that this animal laid down, made his bone out of 230 million years ago. Because that's not mineralized. It's still a collagen. So I think that's pretty cool. Next.
But no DNA.
No DNA. No, DNA is much too. Collagen is very hard to break down. It has such a dense, tightly laminated structure that it is quite resistant even to biological attack by bacteria. So how long does collagen persist in the bones? Well, for a long time, the collagen lasts a long, long time. Back in 1963, they found collagen in Devonian age fossils, 380 million years old. And they positively identified it in Miocene bone, 30 million years old. So collagen itself as an organic molecule can stay under the right conditions, can stay around for most of life's history. Here's a little bit of reprint from the 1963 abstract that they found specimens of Dinichitis, whatever that is, that they could find even in 63 by just using sectioning and cutting it with a microtone. You know, they took all the minerals out of it and they could still cut it up like it was a piece of tissue and stain it and find that it's still there, still a soft tissue. They got polysaccharides and collagen. Pretty interesting. Okay, next. So what's controversial? So we've had a lot of controversy over the last 20-something years. Mary Schweitzer at the Museum of the Rockies claimed to find tissues in T-Rex, and that was really, really controversial. Oh no, it's just bacteria that grew in there. No, if you look at the kind of molecules we're finding, they're not molecules made by bacteria. They are molecules made by advanced animals, the type of collagen and things like that. What was controversial is not the fact that there was collagen still left there, because we'd known that for 40 years, 60 years. What was controversial is that they claimed to find flexible structures still made out of collagen, as opposed to the mineralized collagen. And it looks like they were correct. You know, subsequent work, they've done lots and lots of work that validated those claims. There are collagen structures left in bones under the right conditions that are flexible and can be removed. I didn't find any in Bernie's bones. I did decalcify some Bernie bones and look at it and didn't find, well, I found some stuff that might be, we'll look at it, but not very much. So it that there's going to be collagen present in old bones. Those osteons, half of their volume, roughly half the volume, half the weight, is made up of collagen. And if the collagen had been degraded, we would have lost all those structures at that level, at that 200, 300, 400x level. We would have lost structures, but the structures are still there. Okay, next. So here's a couple pictures of Bernie's bones. So I'm kind of switching over to what now a project that I'm kicking off. So here's some structures. These pictures came from Joe at the Fossil Festival. He took my thin sections, which I had made from bones. I didn't make them. I paid somebody to make them over in Vancouver. And these are some overall pictures of the bones. And you see they are actually beautifully preserved. These are all the white here on either bone. That's actually what would be called spongy bone. These bones are very spongy. They do have some hard parts, but most of them are pretty light and spongy, which you could expect that from an animal that lives in the water a majority of the time. But they are very well preserved. Next. So what I'm going to do on this study, I think there are organics present in Bernie's bones, and I want to be able to show it and prove it and talk about it. You know, it's kind of a cool thing to have Oregon's oldest bones still have organics. So some of the evidence of Bernie's bones containing organics are: one, this still has a good structure. The structure at the atomic level or at the 200 to 400x level has not degraded. That tells me that framework of the collagen is still there. We still have osteons. We can see them. They look like immature osteons, not mature, but they're there. If you look at this one right here, this is one of the bones that was exposed on the surface of the road cut that we found. This is one of the ones that led us to the group. I picked it up because it had a nice nautilus on it. Didn't realize till I got it home and cleaned it that it had bone in it. But if you look at the bone is all bleached white. So we have a substance in there that normally is black, is bleached white under sunlight. Most minerals don't bleach. They're inorganic. So that I suspect that it is organic material that both turns black and then turns white. And there aren't any black minerals in the matrix to stain the bones black. So we're going to go through and we're going to scan these sections. We're going to look at the atoms present, the types of transition metals that might make black minerals. We'll look, probably won't be able to actually directly measure carbon because that's very hard to measure with an SEM, and it's just hard to pick up, or an X-ray. They just don't detect very well. I might, if we're lucky, we might find some carbon black or something, some little pieces that tell by the structure, because that might be the color centers. And if it is collagen, I've got some sample set off now to a researcher who's at Ohio. What's her name? I can't remember the name, sorry. That has done a lot of work on looking at the collagen in fossils and see if we can get some type of collagen, because most of it's type 1, what they call collagen type 1, but there are variations within the major animal groups. So we'll see if it should correspond, of course, to a reptile collagen. We'll find out. I haven't gotten any results back from that yet. So let's see, do I have anything else? No, that's it. So that's my talk. We're just kicking this off. If you want to come up and look at the thin sections and thick pieces that were left, you can. This is $500 worth of work, so don't drop them on the floor and step on them. Other than that, they're pretty sturdy. So questions? Any questions? Not that I can maybe answer them. I'm just reading this research. Yeah.
Used acids to remove the inorganics?
I used EDTA. Yeah, okay. The question is, did I use acid to remove the minerals, to demineralize it? Actually, I used a slightly different process. I used, they recommend using EDTA to chelate the calcium and remove it. And that's what they've done on other studies. So I just copied what somebody else did. I did do that. The bone fragmented quite well, left a black residue, which I have collected on little filter pages. I didn't see, I was actually looking for little capillaries, and I didn't see anything under a couple hundred power. except for some stuff that I suspect might be glue, because there was some glue on it. I tried to clean it all off, but there may be something there. But yeah, I used EDTA to demineralize the bones. Yeah, I soaked it in EDTA, took 2 weeks. So nothing you're doing it in its office. Yeah. Yeah, I mean, I could have put it in stronger acids, but I decided not to. I'd follow somebody else's protocol. Yes, Kent.
Wonder, like on a skull where the sutures are on a growing animal.
Yeah, that's a good question. And, that. Yeah, it is interesting that well, you teach anatomy. Did you answer that question? So it has the right. It has the right. That growth hormones cause the growth or stop the growth, but what controls the growth hormones? We can go back and back. It gets back into what we do not understand. I certainly do not understand. You maybe understand. No, a little bit. So yeah, I don't know why, but I do know that, when a bone stops, when animal stops growing, that the growth plate fuses to the rest of the bone, and eventually it gets reworked to the point that there is no line left in that. In a fully mature individual, you can't tell probably that there was a growth plate there. It's just solidly fused all the way through. One other interesting comment, and this is pretty speculative. Pat Druckenmiller up at the University of Alaska has a lot of these good bones from Bernie that we prepared. This stuff is just scrap that was left over at OMSI, which is great because I've still got lots and lots of scrap to play with. But he sent off some of the bones to a researcher in Switzerland. And that researcher in Switzerland compared the bones of a Swiss thalattosaur, which some of the early thalattosaurs were found on the Swiss-Italian border, which used to be a shallow sea in the Triassic. Kind of hard to believe, but yeah. So they have Swiss thalattosaurs as well. And so he compared the bone structure, the osteology of the bones from Switzerland, Italy, to the bone to Bernie's bones. Even though that classification paper has not been written, they just referred to it as the Oregon thalattosaur. So everybody knows there's a thalattosaur coming out of Oregon. And one of the interesting things that the guy noted is that none of the bones he studied, and he had 4 of them sectioned, none of them are mature. They are all what I saw, like on this one, I didn't really picture it real well, that it does not have that nice laminar, I won't say spiral, concentric design. Does that mean he attributed maybe a different lifestyle? Develop that, or that might be something peculiar to the family of thalattosaurs. The bones from doesn't even show that kind of stuff. They're really bad shape. The other alternative, which I'll throw out to you guys, and I think I've talked to some people about this before, we found one bone or one shaped bone in the thalattosaurs. It's called the chevron, and it goes underneath the tail, and it's used to protect the main artery and vein that run back and forth along the bottom. So the neural arch protects the spinal cord that runs along the top, and the hemal arch protects the bottom. Well, we found 3 or 4 of these chevrons, these hemal arches, in the mass of bones. Most of them were about this big. One of them was that big. And if you scale that big one up, then it becomes a 9-meter animal, which is 30 feet long. But it's only part. There's most of it, probably 3/4 of it is there, but there is a little bit missing. And it clearly looks like the ones from the thalattosaurs, but maybe it's from a different animal. And I have no idea what Pat is ever going to say about it. But if that was indeed from a mature thalattosaur from Oregon, then that means all the other bones we have are from immature, still growing juveniles. Even if they're 14 feet long, they're still only half the adult size. And that could also explain why all the bones that have been studied are not mature bones, because they're not from mature animals. So that one's pure speculation at this point in time, but it's kind of cool to think about it, because as it stands, Bernie is the largest thalattosaur found in North America at maybe 14 feet long. And it may have only been half grown. So I'll leave you with that. That's, yes.
Talk about taking in sections of that big chevron.
I am going to ask Pat to send that one back to us so that we can do that. We have not done it yet, but I definitely want to do it. Or maybe have him send it to the researcher in Switzerland who did the other ones. And because we've only got that one bone that would have been from a big one. All the other bones, they're all consistent with this, with the 14-foot or smaller animals. And one other caveat on that is all the other thalattosaurs in the world, the largest one ever found is one in China that's roughly the same size as the one in Oregon. But most of them are only 2 to 3 feet long. So if indeed we have one 30 feet long, that's 10 times. And I'm not sure any professional paleontologist is going to stick their neck out and claim that they found that, but I'd like to. Yeah.
Say, Greg,
Yeah.
Harold Hines online here. This has been a fascinating talk. Learned a lot of stuff. I'd be really interested in you've done a lot of background reading now. Tell us something about the evolution in general of bone structure. What have you learned? When did it first appear and how does it develop, and what kind of studies have been done to show these trends?
I believe, I'm not really sure. I'm pretty sure, I think the first mineralized bones appeared in the Silurian, not too long after the Cambrian. But and I know by the time we got to Devonian, we have fully blown mineralized skeletons in the fish. But I have not really studied that. When did mineralized bones start to develop?
Or their evolution over time?
Yeah.
Harold needs to read a book on it and write a review.
Oh, okay, Harold, you need to tell us. That's what Tara said.
No, it's. I've done a good bit of reading on the recent studies dealing with bone structure and so on, and the great debate as to whether they're cold or warm-blooded, and that's a fascinating literature all by itself. But anyhow, this has been a fascinating talk.
Yeah, well, we know that bones don't matter. It doesn't matter whether you're cold or warm-blooded. you can have mineralized bones. I mean, fish
That's
proved that a
true.
long time ago. No, I can't answer your question. Yeah,
Skulls give mammalian bone similar to amphibians or fish.
very much so. Yeah. Yeah, you burrow down into what the bone structure looks like, and they're very much the same. They have the same chemistry, the same types of cells. They have the same microstructure or mesostructure. Yeah, yeah. We all inherited this from. Well, that would have been back in those lineages split in the Devonian. Well, maybe, yeah, maybe Devonian. That land animals split from ocean animals. Yeah, we go back 380 million years or so. Yeah. And the same way of making bone has persisted. I mean, why do we still have the same number of bones in our arm as the lungfish? You know, one bone, two bones, many bones, digits. Same with lungfish. And that's Devonian was when that occurred. So it's something that it works well, and it's been preserved by evolution. You know, a trait that's been preserved. Okay. Any other questions? Okay, I guess we're done. I guess.